Welcome to today's class on plant physiology and metabolism, where I am going to dive into the study of abscisic acid, another plant growth regulator. Get ready to explore the significance in plant biology. From 1935 to 1965, focused on investigating substances responsible for bud dormancy in woody plants, particularly the sycamore. It is also known as Acer pseudoplatus. Moving forward to 1961, Endicott initiated research exploring compounds that promote leaf abscission, particularly in the early 1960s when pivotal breakthrough unfolded in the realm of plant physiology. In 1966, Liu and Cons achieved a significant milestone by successfully isolating crystals of abscissin from the petiole of cotton plants. This marked a crucial step in understanding the elusive substance involved in plant processes. In 1963, in Japan, Okuma et al. Expanded our knowledge further by isolating abscissin 2 from the young cotton fruits. Simultaneously, during the same year of 1963, Eagles and Waring made a noteworthy contribution by identifying and isolating dormine from Betula pubescens. This substance found in the buds of growing seedling was revealed to induce dormancy. These breakthroughs not only unveiled specific plant hormones but also laid the foundation for deeper exploration into the intricate world of plant physiology and the regulatory mechanism governing growth and dormancy. In the year 1966, abscisic acid was successfully identified from several species of seed plants. Finally, the significance of this discovery was further underscored in 1967 during the 6th International Conference on Plant Growth Substances. At this influential conference, the compound was officially designated as abscisic acid or ABA. Now, coming to ABA occurrence, it is a widespread plant hormone found throughout the vascular plants, and interestingly, certain fungi also produce ABA as a secondary metabolites. In higher plants, ABA is omnipresent, existing in all living tissues from the root cap to the apical bud. Notably, it is synthesized in cells containing chloroplast or amyloplast, showcasing its involvement in crucial processes such as photosynthesis and starch storage. This pervasive distribution underscores the fundamental role of ABA in regulating diverse physiological responses across the entire plant organism. As you look in the figure, ABA is sesquiterpene that 15 carbon atoms from an aliphatic ring system featuring a double bond, 3 methyl groups and a unsaturated chain. This chain concludes with a terminal carboxyl group contributing both dynamism and functionality to the ABA's molecular makeup. Moreover, ABA is present in different other forms also, such as physic acid, alpha abscisic acid and beta abscisic acid and so on. Now let us discuss about the ABA biosynthesis. As I have already told in the previous slide that ABA is a 15 carbon sesquiterpenoid which is synthesized in cells with chloroplast or amyloplast. Its production involves a complex pathway from 40 carbon carotenoids intermediate initiated in plastid. The pivotal and highly regulated step is the cleavage of carotenoid by the enzyme NECD, that is 9-cis epoxycarotenoid dioxygenase. This event produces the 15 carbon precursor molecule, that is xanthosine. So, it means that xanthosine is a precursor for the synthesis of the abscisic acid, which then ventures into the cytosol. In the cytosol, a series of oxidative reactions unfold covering xanthosine into the active ABA molecule. However, the story does not end there. ABA actively can be modulated by the hydroxylases, leading to inactivation. ABA can undergo reversible inactivation through conjugation, both tightly regulated. Both the process is tightly regulated. Now let us talk dynamics of the ABA, that ABA concentration exhibit dramatic fluctuation during development and in response to environmental cues in developing seeds. ABA levels can soar by 100-fold within days, reaching micromolar concentration only to decline sharply as maturation progresses. Conversely, under water stress condition, ABA level in leaf can surge 50-fold within 4 to 8 hours. Let us explore the transport of ABA. Basically, the mobility of ABA serves as a dynamic signal that influences plant physiology through its synthesis and action in dynamic cellular location. The systemic ABA response is intricately linked to the direction and flow rate of ABA transport. As in dehydrated plant, vascular tissues emerge as a key site, traverse to God cell and mesophyll cells, forming a crucial part of ABA's regulatory network. Notably, God cells contribute to this regulatory dance by both synthesizing and degrading ABA finely adjusted cellular ABA level. Shifting next to development, synthesized ABA premature prevents germination. As we progress into the embryos and endosperm assumes a critical role. Here it becomes a key player in inducing seed dormancy and inhibiting the premature germination of the seeds. Now come to the ABA transporter. Now let us delve into the captivating world of ABA transporter. ABA transmembrane transporter play a key aspect influencing physiological response in plants. Recent breakthrough in Arabidopsis research have uncovered two distinct types of ABA transporters, each playing a unique role in the plant physiology. The first one is a ATP binding cassettes, or you can say that the ABC type transporter, and another one is NRT1 PTR family, or you can say that the NPF member protein. As we discussed, these ABA transporter, their activities contribute to the regulation of diverse physiological responses from stress adaptation to developmental processes. Now look forward to the fascinating mechanism through which abscisic acid triggers transformative changes within plant. Firstly, ABA alters the plasma membrane. This transformation is akin to a change in the bioelectric potential across membrane. As a consequence, there is a leakiness that leads to the efflux of potassium ions. This effect holds particular significance in processes such as stomatal closure, where the controlled movement of ions play a pivotal role. Second mechanism shown by ABA is control over cellular processes by inhibiting both RNA and protein synthesis. This involves the suppression of alpha amylase activity and the selective limitation of specific protein synthesis. Moving to next, ABA promotes the inactivation of GA by covering it to glucoside, thereby impeding the functions of GA in processes such as seed germination, dormancy, and growth. Coming to an alternative hypothesis which suggests that ABA operates through the participation of secondary messenger including calcium and calmodulin. Calcium plays a role in ABA-induced geotropism in maize roots. According to Kokoi and Negini in the year 1989, ABA hinders germination by regulating the progression of certain activities that rely on the calcium calmodulin system. The physiological role includes the inhibition of seed germination, inhibit the seedling growth and bud growth, help in the senescence and the abscission. ABA also increase the yield if tubers and stimulate fruit ripening. ABA is called as a stress hormone as it increases stress tolerance in plant. In the seed development, we encounter the phenomena of dormant seeds where growth is temporarily halted. ABA takes center stage in inhibiting the germination of these seeds, acting as a regulatory force influenced by the balance between ABA and GA. The dormancy of the embryo is attributed to the presence of inhibitor, with ABA playing a prominent role while growth promoters like GA are notably absent. This interplay between inhibitory and promoting elements contributes to the state of dormancy. The dormancy may be due to inhibition of enzymes like protease and amylase along with the suppression of carboxypeptidase synthesis. ABA in its multiphased role also exhibit the ability to inhibit water uptake during the germination process. Moving ahead, it is further said that ABA promotes the dormancy of buds by causing the shortened internodes, particularly inhibiting the sprouting of potato buds entirely. ABA also induces senescence and abscission processes in plant, which can be seen from the present figure. Now, we will discuss that how ABA act as an anti-transpirant. The elevated ABA levels contribute to the closure of stomata. ABA hinders the transport of potassium ion from the external environment to the guard cells and release of malate from the guard cells. This results in the flaccidity of guard cell, ultimately leading to the closure of the stomata. As discussed, ABA closes stomata, which is a protective mechanism employed by the plant to reduce water loss through transpiration. By closing stomata, the plant minimizes the exchange of water vapor with the atmosphere, helping to conserve water during period of stress. The closure of stomata induced by elevated ABA levels thus plays a crucial role in the plant's ability to adapt to challenging environmental conditions and sustain optimal water balance. The use of abscisic acid in agriculture offer a broad range of advantages for plant development and resilience to stress. ABA increases the yield of tuber. It also induces flowering in short day plant. Moreover, ABA application delays the opening of flowers and sprouting of storage organs like potato. It also stimulates fruit ripening. As you know, ABA delays seed germination by inducing seed dormancy. Moreover, ABA acts as an anti-transpirant, therefore, it increases the stress tolerance in the plants. As from the figure, it is evident that abscisic acid increases a signaling pathway for alteration in ion concentration, water equilibrium, as well as the ionic changes. The initiation of gene expression ultimately leads. The stomata close and the which leads to the inhibition of seed germination and as well as the growth, which further leads to the stress tolerance. Now, the major thing about any research is basically the signals transduction. So, now we will discuss about ABA signaling in the guard cells. As it is shown in figure, ABA bind to the cytosolic ABA receptor. They are RCAR or PYR or PYL. After the binding of ABA to receptor leads to the formation of a complex with type 2C protein phosphatases or PP2CS, which cause a reduction in their activity. PP2CS play a significant role as a negative key regulator in this signaling network. Then inhibition of PP2C actively release the positive regulator kinase OST1 from this inhibition, resulting in both phosphorylation and activation of NADPH oxidases or RBOS. RBOS catalyzes the formation of apoplastic reaction oxygen species such as hydrogen peroxide and superoxide, which triggers opening of plasma membrane, calcium ion, permeable ion channels. Calcium ion enters the cell. Consequently, the rise in cytosolic calcium ion, further enhanced by the calcium ion, facilitated release of stored calcium ion from organelles like the vacuoles, which initiate the activation of calcium dependent protein kinases, or you can in short say that CPKs. These CPKs in turn have the ability to activate RBH proteins, thereby amplifying the influx of calcium ion into the cytosol. As shown in the figure here, that OST1 and CPK both phosphorylate and activate anion channels located in the plasma membrane. Therefore, the activation results in the efflux of anions. In the absence of ABA, this process can be directly inhibited by PP2CS. There are two types of anion channels that are activated. They are slow or S type and rapid or R type. The efflux of anions results in the depolarization of the plasma membrane, as you can see in figure, causing the outward movement of potassium ion through the outward rectifying potassium channel. In a plant cell, a significant portion of both anions and potassium ions is stored in the vacuoles. These ions are released into the cytosol through calcium activated protease channel and anion release transporters located in the tonoplasm. The outward flow of ions leads to a reduction in turgor pressure within God cells, ultimately causing the closure of stomata. As you can see in figure, therefore, as you can see, the suppression of PP2C by ABA and RCR-PIR-PYL result in the inhibition of plasma membrane channel responsible for the accumulation of ions during stomatal opening. These channels include hydrogen ion ATPases and inward rectifying potassium ion channel. In the absence of inhibition, these channels would typically counteract the effects, promoting closure by facilitating ion efflux. Our discussion on this intriguing subject of abscessic cases comes to an end for today's session. Thank you.